The information in this module is accurate and complete to the best of our knowledge. All recommendations are made without guarantee on the part of the author or the sponsoring institutions. The author and the sponsoring institutions disclaim any liability in connection with the use of this information.
Welcome to the course, Biology of Genomes. Genome biology is a relatively new science that formed on the interface between the domains of several biological disciplines, as well as informatics. In this course, we will address structure, function, organization and expression of genomes, comparative genomics, natural genome diversity, population genomics, and environmental genomic interactions. We will also discuss contributions and insight from the fields of computational biology, structural biology, developmental biology, and cell biology, as well as theoretical advances in the field of genome evolution. This course does not limit itself to a genome of single organism. Instead, it embraces all taxonomic levels and for all forms of life, within populations or across domains. Its aims are to further the understanding of genomes in an evolutionary context, and further the understanding of evolution from a genome-wide perspective. The title of the first lecture is From Genes to Genomes. At the end of this lesson, you should be able to understand what a restriction enzyme is and how it is used in the recombinant technology. Mention an example of medicine that have been produced with recombinant technology. Describe how polymerase chain reaction produces in vitro clones of DNA. Describe the differences between major sequencing methods. Understand what is so special about ddNTPs. The source of genome science can be traced throughout 20th century, all the way to the discovery of the concept of the gene. For a long time, the gene remained an abstract concept, often considered to look like a bead on a string. Only in 1952, the experiments of Hershey and Chase have shown that DNA was indeed the molecule of heredity. This meant that the information in the DNA was connected to the visible phenotype. Not too much later, Watson and Crick made their discovery of the DNA structure. This suggested the connection between the order of nucleotides in the DNA molecule and their function. Indeed, the connection of major disease to chromosomal abnormality was shown soon after, with the example of Down syndrome. However, it took almost 10 years from the discovery of DNA structure until the mechanism of functional connection was firmly established with the discoveries of mRNA and the genetic code. In the 1960s, there were still very few sequences available to study, but the tools required to identify, isolate, and manipulate desired stretches of DNA were already evolving. One of the most important was the discovery of restriction enzymes, proteins that were isolated from prokaryotes that cut DNA at specific recognition sites and can be used to splice DNA from other species. The resulting sequences are called the recombination DNA because they consist of combination between DNA sequences from different sources. With these tools in place, the recombinant DNA age was about to allow scientists to start cloning genes in the test tubes for the first time. The development of recombinant technology signaled the advent of genetic engineering. The recombinant organisms that were created allowed to produce valuable pharmaceutical products on massive scale, reducing their price, such as insulin and human growth hormone, which are used to be very expensive and had to be derived from animal corpses before this. At the same time, it also led to the discovery of in vitro cloning, a technique to quickly make exact copies of DNA fragments, including genes. As we will see, molecular cloning will have huge practical uses as well as scientific implications. In turn, restriction enzymes can be used not only for molecular cloning. One other widespread use of this technique is DNA fingerprinting. This is a laboratory DNA technique that can be used to link the biological evidence left at the site of crime and a suspect in a criminal investigation. First, the DNA is extracted from the cells, purified, and treated with restriction enzymes. Since the enzymes cut in specific places, the treatment produces a mix of fragments with different length. This mixture is then subjected to electrophoresis, creating a unique pattern of profile of each individual. The reason that restriction patterns differ between individuals is that a single mutation can add or remove a recognition site and change the resulting fragment length. On the picture, you can see a restriction pattern produced by a DNA sample taken from a crime scene, compared with a pattern from a suspect. If the two DNA profiles match, then the evidence came from that suspect. DNA fingerprinting could also be used for studying genetic variation. The restriction profile cuts genome into smaller pieces, and scientists started to use a hybridization technique to identify which fragment contained a gene of interest. In a widespread technique called southern blotting, the restriction profile is transferred to a filter membrane and a probe is added that attaches to the sequence that is being studied. Then the rest of the fragments are washed away and the restriction profile only inside of this gene can be visually examined on the film. The probe can be labeled either with radioactivity or fluorescence. A hybridization probe is basically a fragment of DNA or RNA of variable length, usually from 20 to 1000 bases long. It attaches to a complementary sequence in the genome. The technique you see is called restriction fragment length polymorphism, or RFLP. It is a combination of fingerprinting, blotting, and electrophoresis that allows to see variation inside of the gene of interest. Once more, you can see that DNA extracted, cut from the restriction enzymes, and the resulting fragments are separated by the electrophoresis technique. The resulting bands are transferred to a membrane by southern blot, and a radioactive probe is added. This probe will bind exclusively to the fragment containing a complementary sequence. The membrane is developed using X-ray and produces a photograph of the bands. These bands will represent variation in the restriction site within the fragment bound to the probe, or RFLP. A similar concept to a probe is a primer. Primers are short, unique strands of RNA or DNA, generally about 18 to 22 bases that bind to the specific site, serve as a starting point for DNA synthesis. Primers are normally used by the cellular machinery and required during DNA replication because DNA polymerases, the enzymes that catalyze this process, can only add new nucleotides to the existing strand of DNA. In other words, it can only extend the existing strand, not generate a new one. The human genome contains approximately 3 gigabases of DNA. A probability calculation can show that you can find 12 nucleotide sequences approximately 200 times, but 18 nucleotide sequences only 1 in 200 times. Therefore, it is a good chance that we can design primers longer than 18 base pairs that are unique and will only be binding to the specific sites we choose. Primers can allow us to isolate and amplify a specific DNA sequence and study it without incorporation into a living cell, or in vitro. In a polymerase chain reaction, or PCR, two primers are designed to match the opposite ends of a DNA segment inside the genome that we want to copy. We first warm up the solution containing genomic DNA so that strands separate to convert the genome into a single-stranded DNA. This is needed so the primers then can be attached to their complementary sites. Then the temperature is lowered, and through complementary base pairing, one primer attaches to the top strand at one end of the segment of interest, and the other primer attaches to the bottom strand at the other end. The temperature is then brought up to 72 degrees Celsius, perfect for the enzyme polymerase to extend the primers and synthesize the complementary strand. This is done by sequentially adding the dNTPs, or the single nucleotides, A's, G's, C's, and T's, to match the complementary nucleotides on the old matrix strand. After this is done, the temperature is brought up again to separate the old and the newly made strand. By varying temperature in the thermocycler, the PCR produces millions of copies of the target DNA in a matter of hours. There is only a short technical step that separates PCR and DNA sequencing: a direct reading of the order of nucleic acid in the sequence. In the process called Sanger sequencing, which is still a golden standard of sequencing technology today, special nucleotides with fluorescent markers are attached, called ddNTPs. Note the double Ds. They are used in the PCR reaction and in mix with regular nucleotides, or dNTPs, with a single D. When such a ddNTP is incorporated, the chain is terminated, and the truncated product is released with a fluorescent tag. The fluorescent marker indicates that nucleotide is at the end of the fragment. So now we will sort out the fragments in the same way as we did in DNA fingerprinting, by electrophoresis. Just like the PCR, sequencing starts by adding primers and amplifying the target sequence. However, because of the presence of ddNTPs, the sequences will randomly terminate in different lengths. Now, fragments can be subjected to electrophoresis and will align, forming a ladder with different colors indicating a nucleotide present at the end of each sequence. As the fragments pass in the front of the laser, the sequence is read to a computer. In the 1970s, with the advent of first-generation DNA sequencing, it became possible to read the entire sequence of cloned gene with relative ease, perhaps 1,000 to 30,000 base pairs long. In 1990s, capillary sequencers allowed a massively parallel approach that allowed multiple fragments to be sequenced and assembled into a whole genome sequence of an organism. This led many exciting developments in the 1990s, including the Human Genome Project, or HGP, and the beginning of genome science. This concludes today's discussion of the Genome Project. In the next lesson, we'll continue studying this subject.